one of the things that people are really excited about with respect to psychedelics and related compounds, MDMA, ketamine, and so forth, is that there's some evidence that they can be therapeutically useful, and not just useful, but useful in ways that overcome a lot of the shortcomings that a lot of traditional psychiatric medications have. There's evidence that you can just give a small number of doses to see remission of things like PTSD, that you don't need to keep taking it chronically for months and years at a time. So they're fast acting in that sense. They also have the effect or the side effect, depending on your perspective, of they induce subjective effects, hallucinations in the case of classical psychedelics, the stimulant and prosocial effects in the case of MDMA, the dissociative effects for things like ketamine. And there's some debate right now in the field in terms of whether or not the subjective effects are just epiphenomenal or they're required for the therapeutic benefit. Based on your whole career in synaptic plasticity and experience dependent plasticity, how do you think about this?
I think that is perhaps the critical question in the whole field of psychedelic research, and in the context of using these drugs as therapeutic agents. Everybody in the field wants to know the answer to that question. My personal intuition is that it's going to be very difficult, if not impossible, but it's a personal opinion, not based on real data, to dissociate the subjective experience in response to these drugs from their therapeutic efficacy. That doesn't mean, so I really believe, and it's a little tricky. Do you need the trip for the therapeutic efficacy, or is it you can't dissociate them, but you actually don't need the trip? But and as you, so I'm not explaining this very well. As you know, I'm sure there are companies, startup companies, biotechs, working really hard in developing analogs of these classic psychedelics, not only psilocybin, LSD, but including drugs like ibogaine, with the assumption that molecularly they can dissociate. They can make a drug that's, for example, still activates the serotonin 2A receptor, but does not cause a trip, or activates the 2A receptor. And maybe there was just a paper in Nature about the potential importance of a different subtype of serotonin receptor. As I said, my belief is it's going to be pretty difficult to separate the two. We're actually not going to really know the answer until these drugs that biotechs are creating are actually tested clinically. Because we have very simple-minded models in mice to tell us whether a drug is hallucinogenic or not.
Mm-hmm.
But
we ultimately don't know until a human being takes it.
You really don't know till the human being, because the mouse can't tell you what it's experiencing. And let me just think, is there any clinical evidence out? Oh, the point I wanted to make is, so that doesn't mean, let's just say my intuition is reasonably correct. You still need some subjective experience to get the therapeutic action. That statement does not mean there is a lot of negatives there. There is not a lot of room to improve upon the classic hallucinogens in terms of, maybe you don't need to, currently in the clinical trials, let's say for psilocybin or even LSD, the doses that seem to have in early trials seem to have some therapeutic efficacy. You have the subjective experience, that is the trip, for 6 to 12 hours, which means you're sitting, the patient or subject is sitting there in a room with somebody sitting there with them in case they start having what's known as a bad trip. And that's a hard model for the clinical delivery of these substances.
Yeah.
So there's a lot of
scalable.
effort and companies working on, can you shorten the duration of the subjective experience and still get the same therapeutic efficacy? So as you probably know, There are hallucinogenic compounds such as DMT or I always forget methoxy DMT that cause a much shorter trip or subjective experience. And there are companies pursuing those as potential therapeutic agents. There are biotechs, again, figuring out ways of modifying these classic hallucinogens to shorten their duration of action. So there's a lot of room, even if the subjective experience is required, for maybe making these substances more user-friendly in terms of their therapeutic efficacy. But just to be clear, their therapeutic efficacy is far from firmly established. There's a few clinical trials out there that really suggest good effects, but how long the good effects last remains to be determined. And if you actually look at some of the what I would view as the best done clinical trials, it doesn't cure everybody. It's still the case that there's a percentage of patients who don't respond in a therapeutically beneficial manner. So, I'm a believer that these substances absolutely should be studied, and they do have great therapeutic potential, but they need to be studied ethically and rigorously without an evangelical attitude towards them. That's just my personal view.
Mm-hmm. On the opposite side of the spectrum, when we think about the most addictive drugs, things like synthetic opioids like fentanyl, what makes them so addictive? Are they just causing the most dopamine release in those synapses we described earlier?
I know it sounds so simplistic, but that is probably the case. It's a little more complicated when we talk about the dopamine release. There's actually evidence from human brain imaging studies, a lot of it done by the director, not all of it, but some of it done by the director of NIDA, the National Institute on Drug Abuse, Nora Volkoff. And there's actually reasonably good evidence that the addictive liability of a drug correlates pretty strongly with two aspects of the dopamine release in this target structure, the nucleus accumbens. It's the magnitude of the dopamine release, but it's also the kinetics. How fast does that dopamine go up and how fast does it come down? And that's probably the case because it's not only the substance that makes it addictive, it's the route of administration. So one of the reasons smoking meth is actually more addictive than ingesting meth. Because remember, a standard treatment for kids with ADHD is giving them amphetamine.
Yeah.
They're taking amphetamine orally. So that drug has to be absorbed by the GI system. Then it takes, I'm making this up, 10 to 30 minutes to get into the brain. And the kinetics of the drug delivery, let's say an amphetamine taken orally, is it gradually gets into the brain, gradually decays from the brain. So the effect on dopamine levels is very gradual increase and a very gradual decrease. If you smoke or inject methamphetamine, it gets into the brain instantaneously, especially when you smoke it. And I don't know if you're old enough to remember the crack cocaine epidemic. And the reason crack cocaine was so addictive is the route of administration. People were smoking it. It gets into the brain instantaneously. It causes a very rapid, very large increase in dopamine, which disappears over the course of minutes. And that's why if you talk to people who really were smoking meth or crack cocaine, as you know, as a neuroscientist, there's this famous experiment done by James Olds showing that if you put a wire into an animal's reward circuitry and you allow it to press a bar to activate electrically the reward circuitry, the animal will just sit there and repetitively press the bar for hours on end. And smoking crack cocaine or methamphetamine is as close to an electrical stimulation of the reward circuitry as you possibly can get. So you get this very rapid release, this jolt of dopamine, this highly reinforcing event, then it's gone 2 to 8 minutes later. And then what the individual experiencing this is then left with this overwhelming craving or wanting of this drug. And it's a very powerful intermittent reinforcer. And just to go off on a complete tangent here, the gambling industry has paid a lot of attention to the neurobiology of addiction and the neurobiology of compulsive reward seeking. So it's not an accident that, slot machines on a Vegas casino floor, they deliver rewards on a partial reinforcement schedule to make people. I hope this is making sense. So
they're engineered based on our neurobiological knowledge to give rewards on exactly the most reinforcing schedule.
And so, even so, I imagine with a drug like fentanyl, I'm not sure if the imaging studies have been done, but because it's such a powerful opioid, and I think a lot of people smoke it. I believe that's a pretty common route of it.
As far as I can tell, in Seattle, they appear to be doing that.
So it's just like smoking methamphetamine, and maybe even worse. So it's getting into the brain rapidly. So I really think it might be, of course, other things are going on, but it really might be that simple. And it's a little frustrating because we kind of understand the underlying neurobiological mechanism, but that still hasn't allowed us to develop, a treatment that is as effective as we would like.
Mm-hmm.